Hello everyone, my name is Chang and my teammates name are Hà Anh, Thái An and Bach, and we are from Vietnam. Today we want to present our results from the experimental project for the application of guanidine-based polymers in surface disinfection and medical mask production. It is clear for us to see that nowadays the world is facing lots of problems, coming from the pandemic to the long-existing problem of the environment. The development of vaccines and antiviral drugs require a lot of time and effort, especially the safety assessment process. Moreover, the crisis in India in the past showed that the coronavirus is constantly adapting to the environment to create new variants capable of breaking through the vaccine defense. Mask wearing, which we chose today as a non-pharmaceutical approach to protect ourselves from the virus, is actually not the optimal solution yet. Disposable masks, which claim to be the most protective mask these days in terms of antibacterial and antiviral, are not reusable and are being thrown into the environment in numerous amounts. Although people are opting for mask alternatives, for example using face shields rather than masks or substituting disposable masks with cotton ones, these alternatives do not help us to be protective from the virus at all. The numerous disposal of masks into the environment is alarming because of the fact that polypropylene in masks takes a lot of time to be completely degraded, which might result in more environmentally related issues and affect our lives in the future. More importantly, it is the discarded masks that become a place to accumulate and spread pathogens. Therefore, fabricating material that has antibacterial properties, safe to human health, environmentally friendly, with low production cost to cover reusable masks is an urgent social demand.
There are several reasons accounting for our choice of guanidine-based polymers. First of all, compared to popular antibacterial agents such as chloride, which can cause tremendous irritation to our skin, guanidine-based polymers are harmless to humans. Furthermore, they are colorless, have no scents, and can be diluted into water. These polymers can withstand low temperatures too. Most importantly, guanidine-based polymers have antibacterial activities against a wide range of both gram-positive and gram-negative bacteria, as well as particular kinds of virus and mold. Therefore, this project was conducted with two objectives: synthesizing guanidine-based polymers and investigating its antibacterial properties, as well as modifying polypropylene film with guanidine-based polymers and examining its usability. The experimental procedure includes three main steps. First, combine guanidine hydrochloride, hexamethylene diamine, and citric acid in a flask. This mixture is then mechanically stirred at 120 degrees Celsius for approximately 1 hour. During this process, ammoniac is released into the atmosphere. Then the temperature is raised to 160 degrees Celsius and the mixture continues to be stirred for another 2 hours. The most remarkable improvement we made in this work compared to previously published studies was to use citric acid as the catalyst, decreasing both time and temperature of synthesis reactions. In the end, guanidine-based polymers is synthesized. The process of modifying polypropylene film with guanidine-based polymers consists of two steps. First, treat the surface of the film with plasma, then coat the surface of the film with guanidine-based polymers that has just been synthesized.
We use the IR spectroscopy to examine the structure of the product. These peaks have frequency at 2934, 2860, and 1464 wave number, are responsible for the CH bond stretching and bending vibration in the methylene group. Another different peaks at 1644 wave number represent the C-double-N bond vibration from guanidine hydrochloride, indicating that both monomers are well incorporated in the final product. The IR spectrum shows the absence at 3500 wave number, which represents the primary amino group, but instead a broad, strong peak at 3326 and 3180 wave number, which represent the secondary amine. This proves PHMG is successfully synthesized from the reaction between hexamethylene diamide and guanidine hydrochloride. FTIR spectroscopy were used to examine the presence of PHMG coating on PP film. The right figure is FTIR spectrum of PHMG modified PP, while the left one shows the initial polypropylene film. In both spectra, we can find peaks characterized for PP. In modified PP film, there are additional small peaks which are assigned for PHMG. Another difference is the low strain of right diagram peaks, which shows poorer absorbance of the complex product than the pure PP. This difference indicates that PP concentrations is lower comparing to the pure PP. These observations have proved that the PP film, after being treated by plasma, has ability to carry a thin layer of PHMG.
We investigated the antibacterial activity against bactericidal bacterial and fungi. The results as included from Table 1. The as-synthesized guanidine polymers show strong antibacterial activities against both gram-positive and gram-negative bacteria. IC50 and MIC values are smaller than 15 microgram per millimeters and 32 microgram per millimeters, respectively, indicating that PHMG has a broad antibacterial spectrum. Compared to the commercial product, the as-synthesized polymer has obviously higher antibacterial effectiveness. To evaluate the application of PHMG in production of reusable masks, we tested the inhibition zone of 3 samples against E. coli bacteria, BB film coated by PHMG after being washed. As we can see, there is no inhibition zone around BB film, implying BB itself has no antibacterial effect. Meanwhile, both modified BB samples after and before being washed appear inhibition zone with relatively equal radius. It means that BB film coated by PHMG has antibacterial property. Since the antibacterial activities have remained after being washed, the BB film coated by PHMG can be reused for many times. To sum up, the guanidine-based polymer was successfully synthesized and investigated characterization as an antibacterial agent. The experimental results show that the as-synthesized guanidine has strong antibacterial activities against both gram-negative, gram-positive bacteria as well as fungi. The polypropylene film was also successfully modified by the synthesized polyguanidine. The antibacterial investigation indicated the reusability of modified propylene. The antibacterial investigation indicated the reusable of the modified polypropylene film as antibacterial layers in reusable medical masks. Starting from low-cost monomers and simple procedure with low synthesis temperature, but exhibiting excellent antibacterial effectiveness. The as-fabricated guanidine-based polymers had proof in potential for large-scale production. Hopefully, our study contributes an appreciated solution, helping control the current worldwide pandemic.